Genetics is a discipline of biology and is the science of heredity and variation in living organisms. The fact that living things inherit traits from their parents has been used since prehistoric times to improve crop plants and animals through selective breeding. The modern science of genetics seeks to understand the process of inheritance, which began with the work of Gregor Mendel in the mid-19th century. Although he did not know the physical basis for heredity, Mendel observed that organisms inherit traits via discrete units of inheritance, which are now called genes. Genes correspond to regions within DNA, a molecule composed of a chain of 4 different types of nucleotides. The sequence of these nucleotides is the genetic information organisms inherit. DNA naturally occurs in a double-stranded form, with nucleotides on each strand complementary to each other. Each strand can act as a template for creating a new partner strand. This is the physical method for making copies of genes that can be inherited. The sequence of nucleotides in a gene is translated by cells to produce a chain of amino acids, creating proteins. The order of amino acids in a protein corresponds to the order of nucleotides in the gene. This relationship between nucleotide sequence and amino acid sequence is known as the genetic code. The amino acids in a protein determine how it folds into a 3-dimensional shape. This structure is, in turn, responsible for the protein's function. Proteins carry out almost all the functions needed for cells to live. A change to the DNA in a gene can change a protein's amino acids, changing its shape and function. This can have a dramatic effect on the cell and on the organism as a whole. One of the most fundamental technologies developed to study genetics, DNA sequencing, allows researchers to determine the sequence of nucleotides in DNA fragments. Developed in 1977 by Frederick Sanger and co-workers, chain termination sequencing is now routinely used to sequence DNA fragments. With this technology, researchers have been able to study the molecular sequences associated with many human diseases. Researchers have sequenced the genomes of many organisms, using computational tools to stitch together the sequences of many different fragments, a process called genome assembly. The large amount of sequence data available has created the field of genomics, research that uses computational tools to search for and analyze patterns in the full genomes of organisms. Genomics can also be considered a subfield of bioinformatics, which uses computational approaches to analyze large sets of biological data.